Well, thank you all for being with us today and welcome to our August webinar. We are excited to share with you a selection of our preclinical models that can be used to evaluate NK cell targeted immunotherapies. But before we get started, I would like to introduce myself and our expert panelists. So my name is Rebecca Soto and I will be the moderator for today's webinar. I have a PhD in biological and biomedical sciences. My primary role at Biocytogen is to develop scientific content. And I am joined today by our speaker, Dr. Jenna Frame. Jenna is the manager of science communication and marketing and has over 15 years experience working with genetically modified animal models in the field of developmental hematopoiesis. We are also joined by our two expert panelists, Drs. John Charpentier and Jinkong Lin. John earned a PhD in immunology and currently serves as a scientific content specialist at Biocytogen. And lastly, I would like to introduce our Chief Executive Officer, Dr. Lin. Dr. Lin has extensive research expertise in the field of molecular genetics and has held several leadership positions at top pharmaceutical companies and research institutions. And at Biocytogen, Dr. Lin is responsible for the company's US and EU business strategies, external authorization and cooperative development of therapeutic antibody candidates, and gene targeting. And so with that, I would like to welcome our speaker and our panelists. We will be taking questions at the end, so please stay tuned. And now I would like to invite Jenna to begin her presentation.
Excellent. Thank you so much, Rebecca, for that introduction, and good afternoon. Thank you all for tuning in to today's presentation. I'll start today's presentation by introducing the basic functions of NK cells and the types of immunotherapies that are designed to promote NK cell function to promote tumor cell killing. I'll then introduce some of our preclinical mouse models, including our humanized models that can be used to evaluate anti-human antibody therapy, such as monoclonal antibodies, bispecific antibodies, and antibody fragments. I'll then follow up with some data generated in our humanized immunodeficient mouse models, which are geared toward assessing the role of the human immune system in tumor immunotherapies. And finally, I'll provide a brief summary to wrap up before we take any questions from the audience. So natural killer cells are commonly known as NK cells, are immune cells which can be cytotoxic. These cells can induce cell death of target cells through a few different mechanisms, including release of lytic granules into the target cell, through engaging FAS or TRAILR receptors on target cells to induce apoptosis, or through antibody-dependent cellular cytotoxicity, which involves the recognition of the Fc region of certain antibodies, recognizing foreign antigens. And I will further elaborate on this process in the coming slides. So in addition to these functions, NK cells can also modulate immune responses through production of cytokines, growth factors, chemokines, and these can all regulate other immune cell types such as dendritic cells, macrophages, and T cells. And these cell types can in turn also regulate activity of NK cells through the modulation of certain activating or inhibitory receptors. So what are these activating or inhibitory receptors? Well, NK cell activity is generally regulated by these receptor proteins on the cell surface, and they can be divided into two main categories. So we have killer cell activating receptors and killer cell inhibitory receptors. So under normal circumstances, the activating receptors bind to certain antigens on healthy cells to generate activation signals, while the inhibitory receptors bind to MHC1 to ensure that these normal healthy tissue cells are not destroyed. However, in a scenario where the activating receptor is engaged, but the inhibitory receptor is not associated with MHC class 1, the NK cell fails to produce the required inhibitory signals, which causes activation and release of lytic granules to kill the target cell. Similarly, this release can also occur when the activating signals outnumber the inhibitory signals, which can be augmented by things like chronic viral infection or other abnormal effects from things like the tumor microenvironment, again, resulting in killing of the associated cell. In addition, NK cells also express the low affinity receptor Fc gamma receptor 3 or CD16, which can bind to the Fc region of tumor antigen specific IgG1 or IgG3 antibodies and induce cell killing via antibody dependent cell mediated cytotoxicity, also known as ADCC. So before we review the types of models we use to target and evaluate NK targeting immunotherapies, let me start by reviewing some of the general mechanisms of action. So for your traditional monoclonal antibody, the Fab portion will either block or promote the function of the intended cellular target. So here in the case of PD-1, PD-L1, the antibody pembrolizumab serves to block the interaction between the PD-1 checkpoint receptor and its ligands present on the tumor cells. This blockade can help promote T cell activation and anti-tumor activity. Conversely, agonistic antibodies can serve to activate their target in downstream functioning, for example, those targeting the immune checkpoint 4-1BB. So the next category is related to antibodies that have multiple Fab regions, such as cell engagers or bispecific antibodies. And in this cartoon, the bispecific T-cell engager Blincyto serves to engage both CD19 on the B-cell surface as well as CD3 on the T-cell surface. And this particular interaction serves to keep B-cell lymphoma in check. But there are other bispecific engagers that also engage tumor cell antigens in order to specifically activate the immune system. Finally, antibody drug conjugates or ADCs main function is to promote their internalization into the target cell via endocytosis. And when the antibody reaches the endosome, the payload drug is released from the antibody, where it is eventually released and disrupts critical cell functions to result in apoptosis and eventual cell death. So in addition to the antigen-specific Fab region of the antibody, the Fc region of the antibody can also have important functions mediated by various immune cell types. So engagements by the macrophage CD32 receptor can promote engulfment of targeted cells by macrophages, and complement can also be activated to induce formation of the membrane attack complex to lyse the tumor cells. In some cases, complement receptors on macrophages can also recognize this deposition, which can result in phagocytosis. And finally, as I mentioned earlier, antibody-dependent cellular cytotoxicity, or ADCC, can be triggered through the engagement of CD16 on the surface of NK cells. So ADCC can also be triggered by NK cell engagers, which can vary in configuration, but typically are derived from single-chain antibody fragments that can be fused together to enable dual targeting. So many NK cell engagers have dual targeting such that they either target the CD16 molecule to trigger ADCC and or other NK cell activating receptors, which I'll describe in more detail a little bit later, as well as the tumor specific antigen. So in this example, the molecule can target either CD19 and or CD22 on the tumor cells. And finally, CAR-NKs, which are engineered to recognize tumor antigens and trigger intracellular activation, are gaining popularity as a potential alternative to CAR T cells due to the feasibility of the off-the-shelf therapy and reduced potential for long-term effects. So to summarize the broad potential of NK cell-based immunotherapies that I just introduced, these can be divided into a few different categories. So with regard to immune checkpoint therapy, these antibodies can either serve to relieve those inhibitory signals on NK cells or promote activation of NK cell activity. And just to illustrate that a little bit more in a little bit more detail, this slide highlights a short list of the popular immune checkpoints and receptors that can be targeted on NK cells. So in this example, targeting the inhibitory checkpoints or a combination of these checkpoints can help to prevent NK cell dysfunction and promote their activation. And these are summarized here as a list of the inhibitory checkpoints. And on the right side of this chart, there are several activating checkpoints where agonistic antibodies could be employed. So there are multiple antibodies targeting these checkpoints currently in clinical development at various stages. And today we will focus on models to test therapeutics targeting NKG2A, TIGIT, and PD-1. The next set of approaches involves tumor targeting antibodies or bispecific antibodies that can also bind to the CD16 Fc receptor, causing elimination of tumor cells via ADCC. And just to illustrate this with an example, there are tetravalent bispecific cell engager molecules that have been designed to target either CD30 or EGFR, as well as CD16 to induce ADCC. And there are several of these in clinical trials. So in this cartoon, AFM13 is shown, which targets CD30 and is currently being used to treat CD30 lymphomas and other diseases. And finally, the last therapeutic strategy is cell therapies, which can also be evaluated in our mouse models, and I'll discuss a little bit later. So how do we choose an appropriate model for evaluating these novel therapeutics prior to clinical development? Well, it depends on the antibody or the therapeutic type, the target or targets involved, and the expected mechanisms of action. So before I jump into these preclinical animal models, I'll take a quick step back to just provide a little bit more context on what our company provides. So since 2009, we've been providing genetically modified animal models for preclinical research applications. So for antibody drug discovery, we have specially designed mouse models to facilitate each stage of the discovery process, which we refer to as biomice. So our gene targeting platform can be used for target identification and validation using custom GEMM models. And for antibody discovery, we recently introduced our fully human antibody mouse, RenMat, which contains the entire human immunoglobulin variable domain. And this mouse is a very powerful model along with related models for fully human antibody discovery and hit generation. And then after these antibody hits are generated to your target, we offer various off-the-shelf models for later stages of antibody drug discovery, which I'll be focusing on more today, including humanized mice for immuno-oncology targets, cytokines and cytokine receptors, as well as a human immune system reconstituted mouse models that are engrafted into immunodeficient mice. So together, with our pharmacology service platform, we can really support your discovery from target identification all the way to IND. So just to kind of further stratify those mouse models that I mentioned, so for oncology applications, our off-the-shelf animal models can really be divided into three categories to support your tumor efficacy studies. So our Syngeneic models include more than 200 strains of humanized target mice for one or more immune checkpoint genes, common TAAs, and cytokines, and so on. And these are implanted with, excuse me, humanized murine tumor cell lines, of which we have more than 100 different humanized lines to support your efficacy studies, with some examples shown. And then our xenogeneic models involve engraftment of human tumor cells into our highly immunodeficient NDG model and their second generation strain, which I will introduce in more detail later. And thirdly, to evaluate the role of the human immune system in tumor therapies, we offer various human immune system reconstituted immunodeficient models that can then be engrafted with human tumor cells. So first, I will introduce our Syngeneic models. And so I'd like to begin with this first efficacy study example. We used mice engineered to express the extracellular portion of human PD-1, which is, of course, expressed on T cells in addition to NK cells. So these mice can then be engrafted with MC38 murine colocancer cells expressing humanized PD-L1 on the cell surface. And this system allows us to test the efficacy of human antibodies that disrupt the human PD-1, PD-L1 interaction in vivo. So as you can see here on the right-hand side, blockade of either anti-human PD-1 or anti-human PD-L1 results in significant prevention of tumor growth in a robust dose-dependent manner. with the highest doses, which are shown here by the red lines, having the most significant effect on tumor growth. So another important NK cell specific immune checkpoint is the NKG2A CD94 complex, which recognizes the HLA class 1 peptide complex and therefore plays an important inhibitory role in cancer cell killing. So as mentioned earlier, there are several anti-NKG2A antibodies that serve to disrupt this interaction that are actually being evaluated in clinical trials. So to provide a model to test novel therapies targeting NKG2A in vivo, we generated mice expressing humanized NKG2A as well as its co-receptor CD94. And whenever we generate new humanized models, we typically perform preliminary expression validation as well as phenotypic analyses to ensure that these mice have a normal immune system. So here we performed phenotypic analysis using flow cytometry of spleen cells and just to assess normal distribution of NK cells, T cells, B cells, dendritic cells, granulocytes, and macrophages. And what this reveals is really no significant differences in the humanized mice compared with their wild type counterparts. So once we confirm that these mice have a normal immune system, we then go on to perform antibody efficacy studies. So in this study, we used MC38 tumor cells engineered to express human HLA-E. So as you can see here from the green bars compared with the black bars, mice treated with the higher dose of the NKG2A antibody monalizumab analog experienced slower tumor growth, indicating that the antibody was efficacious in these mice at this higher dose. In addition to NKG2A and as well as PD-1, another inhibitory checkpoint that can be targeted on NK cells is TIGIT. So in order to test novel TIGIT therapeutics, we generated humanized TIGIT mice. So in this flow graph is just an example of where we validated human TIGIT expression, shown here in the red box. And as you can see, there's no, the human expression is exclusively restricted to the humanized TIGIT model. So after validating these humanized TIGIT mice, the anti-TIGIT antibody efficacy studies were performed. Antibody treatment of these mice harboring MC38 tumors showed varying degrees of efficacy depending on the specific antibody and dose provided, with no significant health effects such as weight loss apparent. Thus, these studies demonstrate that these mice provide a powerful platform for evaluating efficacy of anti-human TIGIT therapies. So in addition to targeting inhibitory receptors, targeting activating receptors can also be of therapeutic value. So NKG2D activation can be sufficient to trigger NK cell cytotoxicity and result in cytokine production. But in addition to functioning on NK cells, NKG2D can also act as a co-stimulatory receptor on CD8 T cells that can trigger activation alongside TCR activation. So this makes NKG2D a potentially valuable target. And we have recently introduced NKG2 mice to our catalog. So in this model, again, the extracellular domain of NKG2D was the mouse, the human NKG2D was replaced in the mouse extracellular region. And so here you can see validation of human expression in both NK cells, shown here, as well as T cells isolated from the homozygous humanized mice. And stay tuned for future data available in this model. Another new product that has recently been introduced is our humanized NKp46 mice. Which can serve to test new therapies that engage this activating checkpoint NKp46. As I mentioned earlier, briefly, NKp46 has been targeted by these multifunctional NK cell engagers targeting tumor antigens to help trigger tumor cell destruction. In our humanized NKp46 model, you can see nice expression of human NKp46 here in the homozygous humanized mice. And stay tuned for future data available in this model. The next key set of models to introduce for NK cell therapeutics is our humanized CD16A models for testing induction of ADCC potential through the engagement of CD16A, which is shown here. So typically when we humanize our models, we retain the murine regulatory elements. However, in this case, since human CD16A and the mouse version have different expression patterns, we also knocked in human CD16A regulatory elements. So shown here is some expression data indicating, validation of human CD16A expression in NK cells and monocytes, but not in granulocytes. And this is really this really tracks with what we would expect based on our targeting strategy. In this next study, we performed efficacy assessments for CD16 based antibodies in our humanized CD16A mice, and this strain is on the C57BL6 background. And these studies revealed varying levels of anti-tumor efficacy of the few antibodies tested, with no significant changes in body weight evident, indicating that there's no adverse effects. So together, these studies indicate that these models can be used to effectively assess antibodies that target human CD16A. In addition to the models that I just described, we offer other humanized immune checkpoint models for popular checkpoints that are also on NK cells, including LAG3, TIM3, and 4-1BB. These humanized models can then be engrafted with the humanized cell lines, as I mentioned earlier, for efficacy studies that involve disrupting these key checkpoint interactions between NK cells and tumor cells. So some of the most relevant models and cell lines are listed here, but for the full list of models that we offer, please head over to our website or reach out to us to see what products are available or may be in development for your targets of interest. So in addition to our syngeneic models expressing human immune checkpoints on murine immune cells for your initial efficacy validation studies, our xenogeneic models allow for engraftment of human cells into our highly immunodeficient BNDG model and their second generation strains. So these models can be very valuable tools to assess the role of human immune system derived NK cells in targeting human tumor cells. So there are several different strains of human immunodeficient mice on the market, and I'll briefly introduce two here that are relevant to today's topic. So the CV17 SCID model has moderate levels of immunodeficiency with no B cells or T cells, but does have myeloid and NK cell presence. And the BNDG strain is our version of a severely immunodeficient model that has a combination of genetic alterations listed here, including a knockout allele of IL-2 receptor gamma, which results in a complete loss of functional murine B cells, T cells, and NK cells. So the first model that I will introduce that is relevant to CD16A targeting is our humanized CD16A mice on the CD17 SCID background. So since these mice still have murine NK cells, this could be an ideal model if you're looking to evaluate whether your test article is effective at promoting human CD16A activity against human tumor cell lines. So shown here is a few examples of popular human cell lines that have been demonstrated to engraft and grow in this model, including the HCT116, K562 leukemia, and NCI-NMU7 gastric carcinoma cell line. For studies where it could be beneficial to evaluate the effects of your test article on NK cells that are completely human, our severely immunodeficient mice would be your model of choice. So our parental immunodeficient model, BNDG, is similar to those offered by other providers in terms of the mutations that they have. Compared to non-SCID mice, BNDG mice have a longer lifespan and a minimal incidence of thymic lymphoma. They essentially have no lymphocyte leakage. As you can see here from representative data measured by flow cytometry, there are absolutely no B, T, or NK cells present. And this really allows for efficient engraftment of cell line derived or patient derived xenografts for tumor studies. However, one of the challenges faced when we use immunodeficient models with reconstituted human PBMC or CD34s is the lack of NK cells that readily differentiate and function normally in these models. And in particular, PBMC reconstitution is typically dominated by T cells. So to solve this problem, we have introduced humanized IL-15 into our BNDG mice, which again is to promote the differentiation of NK precursor cells into mature NK cells and to really promote the survival and persistence of these cells. So in these humanized immunodeficient mice expressing human IL-15, here we have some validation data showing robust expression of the human IL-15 by both PCR as well as ELISA in the serum, indicating successful generation of the model. In addition to confirming expression of human IL-15, we also performed some proof of concept studies to show that the humanized IL-15 strain better supports human NK cell engraftment and maintenance. So in this study, PBMC derived NKs were sorted and engrafted into either BNDG mice or the BNDG humanized IL-15 mice, which is shown by the red dots, and the parental is shown by the black dots. So as you can see here, NK cells were able to better persist over time in the humanized IL-15 mice, measured by both the cell counts as well as the proportion of NK cells in the CD45 population. And if you're interested to know this, NKT cells do persist in this model. So this would be of significant value. So while PBMCs, reconstitution of PBMCs can be useful for NK and T cell experiments, reconstitution is often biased toward T cells. However, if you engraft CD34 HSCs into these mice, they can also provide other immune cell types that may be of interest for your study. So as you can see from this reconstitution data, when we inject CD34 into BNDG mice, that can really also support better engraftment of B cells and modest levels of myeloid cells that can persist after injection. So once engrafted with CD34s or NK cells, BNDG humanized IL-15 mice can be used to test efficacy of anti-human therapeutics that induce ADCC, immune checkpoint blockade, even bispecifics or combination therapies, as well as CAR-NK efficacy. So in this example study, BNDG humanized IL-15 mice were reconstituted with CD34 cells for 6 weeks and then subcutaneously injected with human A459 adenocarcinoma cells with humanized Claudin 18.2, which is a popular tumor-associated antigen. After 7 days, mice were treated with the anti-Claudin 18.2 analog antibody, and this was shown here by the red curves as compared to the IgG control shown in black. So on the right side here, you can see presence of CD56 positive NK cells as well as CD3 positive T cells present in the blood in the tumor. It's extracted from these mice at the endpoints, indicating this model can be a powerful tool for evaluating role of both NK cells as well as T cells in slowing tumor growth. Our BNDG model, as I mentioned earlier, is also valuable for assessing efficacy of cell therapy, including both CAR-NK and CAR-T efficacy. So while we have performed many CAR-NK studies in-house, unfortunately, I don't have a shareable NK study to show today. But in lieu of that expertise, I wanted to highlight a CAR-T study to stress the capability of these models to evaluate cell therapies. So in this example, Raji-B lymphoma cells expressing luciferase were transplanted into BNDG mice, and anti-human CD19 targeting CAR-T cells were infused 2 weeks later. So bioluminescence was assessed at these indicated time points post-infusion, and you can really appreciate that the CAR-T cells were able to substantially prevent tumor cell amplification in these mice. So before we move on to the Q&A session, I'll first provide a brief summary of the models that we've covered today. So as I mentioned, there are several different types of preclinical animal models to support assessment of novel human NK targeting therapies. And while each one has advantages that depend on your target, type of antibody therapeutic, and mechanism, we've provided some guidelines in this chart, and we are really always happy to discuss experimental details with you as you plan your studies. So for specific NK cell checkpoint targets, we recommend our target-specific humanized models. However, in the case where we don't offer the specific humanized model for your target, or if you're testing a unique combination therapy, bispecific, or cell engager, the CD34 positive reconstitution model in the human BNDG IL-15 mice would definitely be the way to go. For test articles in inducing ADCC, we offer a lot of different options covered today that would allow you to target human CD16A in vivo. And finally, for CAR NK assessments, the CDX model in the BNDG humanized IL-15 mice would be the optimal model for your study. So beyond the efficacy studies in animal models, we also provide comprehensive pharmacology services to help you make the jump from preclinical to clinical studies. So for more information about these other in vitro or ex vivo studies, as well as disease modeling, please contact us and we'll be happy to discuss our offerings further with you. So with that, thank you very much for your time today, and we look forward to working with you in the future.
Awesome. Thanks, Jenna, for that very informative presentation. It looks like we have some questions already in the Q&A. So how are the BNDG mice different from standard NSGs? Are these also IL-2 or gamma gene knockout from non-SCID mice?
Yeah, absolutely. This is quite a common question. Although these are very similar mutations, our BNDG model is actually a complete knockout of IL-2 receptor gamma. So although the genetic targeting is similar, they are slightly different. But empirically, both models should support robust engraftment of human cells. Great.
Okay, so the next question is related to the IL-15 humanized mouse model. So the question is, I assume these human IL-15 mice are soluble IL-15. Have you considered creating trans-presenting IL-15 mice for better NK development and persistence?
Yeah, I mean, that's a great question. I don't know if anyone, if Chin-Kong, if you want to comment whether, we've considered development of this particular model.
Yeah, that's a great question and great idea. And definitely we can consider on that one, but at the moment, I don't think we have it.
Mm-hmm. Yeah, thank you.
Okay, so when we talked about the CD16 mice, we talked about it being on the BALB-C background. And the question is, why is it on the BALB-C and not on the C57 Block 6?
Yeah, so we offer several different strains. So please come talk to us about which model would be most suitable for your studies. But we try to offer different strains to allow for engraftment of different types of tumor cell lines.
Okay, and it seems like people enjoyed the presentation because they want to know if it'll be available for download, and it will be. We will share that link with y'all. We have another question that in terms of graft versus host disease, is it similar level seen in NSGs upon PBMC reconstitution?
Yeah, so we do have another model, another BNDG model that has been engineered to have a B2M-KO, which is supposed to allow for reduced graft versus host disease. But even without that, I think that the length of these studies can persist for several weeks. I don't know if anyone else has anything else to add.
Actually, Gina, the beta-2M knockouts and within our additional engineering, the model is better for the antibody study and also reduce significance of the GVHD to expand the selective window. Simultaneously, we extend the antibody half-life in beta-2M knockouts of BNDG as well. So talk to our team and we can share very detailed information on that model, how to use it.
Yeah, so I think to follow up on that, the next question is for BNDG mouse models, in particular for PBMC or CD34 positive engraftment, would these mice require radiation first? But yes, to Jikong's point, we can definitely work with you and answer any questions that you may have in regards to this particular model.
Yeah, in terms of the point about irradiation, I don't believe it's required. You know, there's obviously improvements seen with preconditioning irradiation before PBMC or CD34 cell engraftment, but much like the NSG model, I don't believe it's strictly required.
Right. Okay. And so next, how many NK cells per microliter of blood are physiologically relevant? And do you see similar or higher levels in BNDG IL-15 mice? Also, have you confirmed the functionality of these NK cells in the IL-15 mice?
Yeah, that's a great question. I think that have we confirmed the functionality? I think we would need to talk to our team whether we've done detailed in vivo studies. But I think the levels of IL-15 are a little bit higher than the physiologic IL-15, if that answers your question.
You know, it's very good questions in terms of how many NK cells in the mice, in these mice, and this is a good question. We know we increase the NK cell, but the exact number we need to find out. And the bottom line, like the question is whether we have been validate the functionality. I think that is the answer. Yes, we did a lot of NK CAR study using this model. So feel free to reach out to us.
Yeah, so similarly, this follow-up question, and so for the BNDG IL-15, the NK cells persist up to 6 weeks after reconstitution. Do you think this study duration is enough for oncology studies? And does this model experience graft versus host like related issues?
Yeah, I mean, that's one of the limitations of these models is that, you know, GVHD-like symptoms do creep up after several weeks. And I think it really depends on the type of tumor that you're targeting and the type of study that you're designing. But yeah, unfortunately, you know, the study cannot go on for months. But yeah, does anyone else have anything to add?
So usually in the for oncology study, we cannot afford 5 to 6 weeks should be good enough. And for some really aggressive tumor models, we can do really small design in terms of the when to inoculate the tumor cells, and that can be discussed very well. So sometimes for some treatment, we may need to do earlier in terms of what time to do the inoculation, what time to do the treatment, and that combination can take care of the therapeutic window issue. Well.
Okay, great. And so some other questions about the IL-15 mouse model. They want to know how high is the expression level. If you're interested, we can definitely share our data package with you, which has some of that expression analysis. So we'll be happy to if you just reach out. The next question, how does the NK/KR repertoire look over time? Is there any selective process or specific education occurring in the CD34 positive HSC model?
Yeah, I think for those detailed phenotypic analysis, we've certainly performed that, and we can share that immunophenotyping with you if you would like to learn more about that evolution over time.
Okay, great.
Rebecca, I think we may have missed a question here from Ram just above Peter's about PBMC-based models and HLA matching. I can take that if you like.
Okay, great.
Yeah, so I would say in response to this that you know, there are protocols for ways to do it, unmatched and matched. HLA matching may give better results. And then to that point, I would also point to other Biocytogen models in which we have transgenic HLA expressing mouse models that might be well suited for this purpose. But yeah, great question.
Yes, thank you for noticing that, John. And so next question is, what's the difference between NSG IL-15 and NCG IL-15 mice?
Yeah, I'm not familiar with how the NCG IL-15 mice was generated, but maybe we can, speak after the presentation.
Mhm. Great. Okay, so yes, another IL-15 question. I believe we already answered that one. Is there any selective population enrichment with IP injection of PBMCs rather than IV?
That's a good question. I don't think we have data speaking specifically to that point, but Ram, if you want to get in touch with any of us after the webinar, we can get back to you with a more specific look at that to see if we've done it before.
Yes, and there's a lot of great questions. We'll try to get through most of them, but if we don't, feel free to reach out and we'll be happy to answer any questions you may have after the webinar. So we'll take a few more, but you mentioned that you engrafted the BNDG IL-15 mice with PBMC derived NK cells and saw good results. Can you engraft with whole PBMCs and still see IL-15 expansion of NK cells in vivo?
Yeah, yes, definitely. We have a lot more data for this model, and we'd be happy to share that with you.
Okay, great. Okay, we have some other questions about specific humanized mouse models in terms of cost. Again, feel free to reach out and we'll be happy to discuss with you any of our available models. So next question is 6 weeks sufficient time to study NK cell exhaustion in oncology studies?
I think this is similar to the last question we had, kind of touching on this, in that, yeah, it's a good point that you're always making a trade-off, and you're always trying to balance, the durability of the study and seeing the effect that you're interested in, with the feasibility of that the model provides. So, is 6 weeks sufficient time? Is a good question, for investigators. But I think that does the model system as it currently exists allow for study of exhaustion? Yeah, I think that's, it's certainly possible. And the duration of the study is going to very much depend on, the protocol that's followed.
Okay.
And John, actually, it's a very good question, very, very professional question in terms of the exhaustion. And then particular activations of NK cell will downregulate their receptors. Therefore, efficacy sometimes is very challenging regarding NK compared with CAR-T or so on and so forth. So it's a good question, but definitely that is the nature of NK cells.
Okay. Do you have experience with co-injection of PBMCs together with tumors versus the systemic injection of PBMCs in the BNDG IL-15 mice? And how about the NK reconstitution and function and GVHG associated with each method? So I think we can approach maybe the first part of that question first. So do we have any experience with co-injection of PBMCs together with tumors versus the systemic injection of PBMCs and IL-15?
I think this is a very similar question we have been common on in terms of when to do the inoculations of tumor, when to do the immune cell constitution. It depends on the tumor model. And I think for the different client, they will ask at different time points. So we do have that one, but unfortunately, we don't have our own data to share on that plan or scheme.
Right. And so to kind of follow up with each methodology, how about the NK reconstitution and function? But yes, if you're interested, feel free to reach out. We can definitely talk about the different methodologies if you're interested. Can the models be used to measure Fc receptor mediated NK recruitment with vaccine antibodies collected from NHP or humans? If so, which model and what is the readout?
This sounds like something that we could probably, plan in consultation with our staff scientists and project leaders. If you wanted to, again, not to sound like a broken record, but get in touch with the specifics of what portions you're interested in having us do, and I'm sure there's a way in which we could help for sure, be that with flow or other instrumentation.
Yeah, absolutely.
Okay. And we'll take just a few more, but how does endogenous ADCC look like in tumor-bearing BNDG mice when dosed with antibodies such as trastuzumab?
Gina may not get your questions. So,
yeah,
what's
I'm
your
looking
question
for
again?
it. Sorry. Go ahead.
Yeah, it's towards the bottom, but I can read it again. So how does endogenous ADCC look like in tumor-bearing BNDG mice when dosed with antibodies such as trastuzumab?
Ching-Kong, were you
I
going
don't
to
think,
feel?
Gina, I don't think we have been paid close attention. Usually when we do the efficacy, we see it in general efficacy. We don't really differentiate the ADCC function or antibody function directly. And people have been using the FC mutation a lot to differentiate the ADCC function was the antibody itself, native function. So I think that need to be compared. Otherwise, sometimes some people use the in vitro to address the ADCC question for your antibody, and in addition to the in vivo. So I think different combination need to be designed very well. But a good question.
Yeah.
I agree with Ching-Kong that, this is something that we can reasonably infer from many, many different studies we've done internally or for clients. We often don't look specifically at mechanisms that closely, but we do see evidence of them happening and working.
Okay, great. And so I'll just ask one more question, and then I think we'll wrap up this great Q&A session. So is there any thought to add the human Mike A/Mike B in the human NKG2D strain, allowing the mice to be to tolerate MICA and Mike B? So I think this is a development question. And as Jenna mentioned earlier in her talk, that we have a lot of mouse strains in development. So if you're interested, definitely feel free to reach out and we can work with you. I don't know if y'all have any other follow-up to that.
I'm not aware of this particular, whether this is particular antigens are in development or not, but we can certainly check.
Okay, well, great. Thank you to our speaker, Jenna, and to our panelists, and to all of your great questions in the Q&A. If we weren't able to answer your questions, please reach out to us via email or our contact us page, and we'll be happy to get back to you. But thank you so much.
Thank you.
Thank you,
everyone.
all, and thank Gina and thank Rebecca and John. Excellent job. And I saw a lot of friends' name in the list. So feel free to reach out to me directly if you have further questions or further need, and a lot of suggestions for us, like what model we should generate for your need, because I saw a lot of experts in NK cell in CAR-T field. So welcome, please give your recommendation. We are a very strong team to generate different models, cell line model, and we will do that one for you. You don't need to pay that. Don't worry about it.
Okay, great. Thank you so much.
Thank you.
Thanks again.
Thank you.
Bye-bye.
Good job. Thank you. Bye.